Imagine we've invented a time machine that takes us back 5,500 years. We embark on a journey through the mysterious world of the past to trace the origins of one of humanity's deadliest diseases, malaria. Yes, you heard that right. This deadly disease has been affecting humanity not just today, but thousands of years ago. What are the origins of this disease? What can ancient DNA tell us about it? Let's dive into this fascinating discovery together. Malaria is one of the deadliest diseases in human history. Even today, nearly half of the world's population lives under the threat of this parasite. According to the World Health Organization, there are about 250 million malaria cases each year, causing over 600,000 deaths. However, the origins of malaria and how it spread throughout history remain a mystery. To understand the depth of its impact and its journey through time, we need to delve into ancient records and cutting-edge scientific discoveries. Megan Michelle and her team at the Max Planck Harvard Research Center have brought to light 5,500-year-old malaria infections using the latest advancements in ancient DNA technology. By examining pathogen traces preserved in human teeth, they have uncovered how malaria spread in the past and influenced human evolution. Michelle explains, "Malaria's legacy is written in our genomes. Genetic variants responsible for blood disorders like sickle cell disease persist because they offer partial resistance to malaria." This statement highlights the intricate relationship between human genetics and disease, where even harmful genetic conditions like sickle cell anemia have been perpetuated because they provide a survival advantage in malaria-endemic regions. How did the two deadliest malaria species, Plasmodium falciparum and Plasmodium vivax, spread globally? Ancient DNA research sheds light on this question. Scientists from 80 institutions reconstructed ancient Plasmodium genome-wide data from 36 malaria-infected individuals spanning 5,500 years across 5 continents. These findings allow us to understand malaria's global spread and its historical impacts on different scales. The ability to trace the genetic footprints of these pathogens through millennia offers us unprecedented insights into the history of human diseases and their epidemiology. In the Americas, malaria is endemic in tropical regions today. Scientists have long debated whether P. vivax, a species adapted to temperate climates, arrived via the Bering Strait with the continent's first inhabitants, or was brought over by European colonizers. Analysis of DNA from an individual found in Laguna de los Condores, Peru, shows a strong link between the P. vivax strain there and ancient European strains, suggesting that European colonizers brought this species to the Americas. The story of malaria in the Americas is not just a tale of disease, but also one of conquest, colonization, and the profound impact of human migration. When Europeans arrived in the New World, they brought with them not only their culture and technology, but also their diseases. Malaria, alongside other infectious diseases like smallpox and measles, played a devastating role in the lives of indigenous peoples. Historical records indicate that entire populations were decimated by these diseases, fundamentally altering the demographic and cultural landscape of the Americas. One of the most compelling aspects of the Laguna de los Condores find is how it illustrates the rapid spread of malaria in the wake of European contact. The genetic analysis showed remarkable similarities between the P. vivax strain in ancient Peruvian remains and those in Europe, underscoring the speed and extent of the disease's dissemination. This finding suggests that within a century or so of European arrival, malaria had already become entrenched in the Americas, shaping the lives and health of its inhabitants. Moreover, the impact of malaria in the Americas went beyond mere infection rates. The disease exacerbated the effects of warfare, enslavement, and population displacement during the colonial period. Indigenous communities, already weakened by conflict and forced labor, faced mortality rates as high as 90% in some regions due to malaria and other introduced diseases. This massive loss of life led to the collapse of entire societies and the transformation of social structures. The legacy of malaria in the Americas is a testament to the complex interplay between human activities and disease ecology. As European settlers expanded their territories and established plantations, they created environments that were highly conducive to malaria transmission. The clearing of forests for agriculture, the creation of irrigation systems, and the establishment of settlements all provided ideal breeding grounds for Anopheles mosquitoes, the vectors of malaria. In modern times, the study of ancient malaria DNA from sites like Laguna de los Condores provides valuable insights into the evolutionary history of the parasite and its interactions with human hosts. By understanding how malaria adapted to different environments and populations over thousands of years, scientists can better predict and combat future outbreaks. This research also highlights the importance of preserving and studying ancient human remains, as they hold the key to unlocking the mysteries of our past. In Europe, military movements played a significant role in malaria's spread. The cemetery at St. Rombout's Cathedral in Belgium, next to one of the first permanent military hospitals, 1567-1715 CE, reveals how soldiers from different regions spread the disease. DNA analysis shows that more virulent cases of P. falciparum were found in non-local male soldiers recruited from the Mediterranean during the 80 Years' War. Alexander Herbig from the Max Planck Institute explains, large-scale troop movements played a crucial role in spreading malaria during this period. The story of malaria's spread in Europe is deeply intertwined with the continent's turbulent history of warfare and political upheaval. During the early modern period, Europe was a patchwork of warring states, each vying for power and territory. Soldiers moving across vast distances and through diverse climates acted as unwitting carriers of malaria. The disease, which had once been confined to certain regions, found new footholds in the wake of military campaigns. The example of St. Rombout's Cathedral is particularly telling. The cemetery's location next to a military hospital provided a unique opportunity for scientists to study the impact of war on disease transmission. The DNA evidence from this site revealed that malaria was not only present among the local population, but also brought in by soldiers from distant lands. The presence of P. falciparum, a species typically associated with warmer Mediterranean climates in northern Europe, underscores the role of military movements in expanding the geographic range of malaria. Moreover, the conditions of military life during this period were highly conducive to the spread of infectious diseases. Soldiers often lived in overcrowded and unsanitary conditions, with limited access to clean water and proper sanitation. These environments provided ideal breeding grounds for Anopheles mosquitoes, allowing malaria to spread rapidly among troops. The movement of armies across different regions also facilitated the mixing of different malaria strains, potentially leading to the emergence of new and more virulent variants. The impact of malaria on military campaigns was significant. In many cases, disease was a more formidable enemy than the opposing army. Historical records indicate that malaria outbreaks could decimate entire units, leading to high mortality rates and severely weakening military forces. This not only affected the outcomes of battles, but also had broader implications for the course of wars and the stability of nations. The parallels between historical and modern malaria transmission are striking. Today, we continue to see how human mobility influences the spread of diseases. Modern globalization, with its rapid and widespread movement of people, has created new challenges for disease control. Airport malaria, where travelers bring back malaria parasites to regions where the disease is no longer endemic, is a contemporary example of how travel can facilitate the spread of infectious diseases. Understanding the historical patterns of malaria transmission can help us develop more effective strategies to prevent and control its spread in our interconnected world. An unexpected discovery was made in Nepal's high-altitude Chokapani region, where a 2,800-meter-high site revealed the earliest known case of P. falciparum malaria. Despite the region being unsuitable for malaria parasites and Anopheles mosquitoes, ancient DNA shows that long-distance trade routes likely brought the infection to this remote area. Copper artifacts from Mustang's burial chambers prove these high-altitude regions were part of extensive trade networks. The discovery of malaria in Chokapani is particularly fascinating because it challenges our assumptions about the geographical limits of the disease. The cold, dry environment of the high Himalayas is inhospitable to both malaria parasites and the mosquitoes that transmit them. Yet, the presence of P. falciparum in this region indicates that human activities, such as trade and migration, played a crucial role in the spread of the disease. The Kali-Gandaki River Valley, where Chokapani is located, served as a major trade route connecting the Tibetan Plateau with the Indian subcontinent. This trans-Himalayan highway facilitated the exchange of goods, ideas, and, unfortunately, diseases. The ancient inhabitants of Mustang were actively engaged in long-distance trade, as evidenced by the copper artifacts found in their burial chambers. These artifacts, which originated from northern India, suggest that the people of Mustang had extensive trade connections that brought them into contact with malaria-endemic regions. The genetic analysis of the Chokapani individual revealed that he was a local male with adaptations for high-altitude living. However, the presence of malaria DNA in his remains suggests that he or someone he came into contact with had traveled to lower-altitude regions where malaria was endemic. This finding highlights the complex interplay between human mobility and disease ecology. It also underscores the importance of understanding historical trade networks and migration patterns in the study of ancient diseases. The story of Chokapani provides a vivid example of how ancient DNA research can illuminate the hidden history of human health and disease. By examining the genetic traces left behind in human remains, scientists can reconstruct the pathways of disease transmission and gain insights into the health challenges faced by ancient populations. This research not only enhances our understanding of the past, but also informs current efforts to combat diseases like malaria. Today, significant progress has been made in the fight against malaria, but resistant parasites and mosquitoes pose a renewed threat. Climate change and environmental destruction are making new regions vulnerable to malaria vectors. Johannes Krause from the Max Planck Institute says, "Ancient DNA studies allow us to explore past parasite diversity, showing how modern globalization makes malaria-free regions susceptible to reintroduction." The fight against malaria has been a long and arduous journey. Over the past century, advances in mosquito control, anti-malarial drugs, and public health campaigns have significantly reduced the burden of the disease. However, the emergence of drug-resistant malaria strains and insecticide-resistant mosquitoes threatens to undo these hard-won gains. In recent years, there has been a resurgence of malaria cases in some parts of the world, highlighting the ongoing challenge of controlling this disease. Climate change is also playing a critical role in shaping the future of malaria transmission. As global temperatures rise, the habitats suitable for Anopheles mosquitoes are expanding, potentially bringing malaria to new regions. Changes in precipitation patterns, deforestation, and urbanization are altering the ecosystems in ways that can promote the spread of malaria. For example, the creation of new water bodies through dam construction or agricultural irrigation can provide breeding sites for mosquitoes, increasing the risk of malaria outbreaks. Environmental destruction and human activities continue to exacerbate the spread of malaria. Deforestation, for instance, can create ideal conditions for mosquito breeding by increasing the availability of stagnant water. Additionally, the displacement of populations due to conflict or natural disasters can lead to the spread of malaria as people move into new areas where they may not have immunity to local malaria strains. The study of ancient DNA provides valuable insights into the long-term dynamics of malaria transmission. By examining the genetic diversity of malaria parasites from different historical periods and regions, scientists can understand how the disease has evolved and adapted over time. This knowledge can inform the development of new strategies for malaria control and prevention. For instance, understanding the mechanisms of drug resistance in ancient malaria strains can help researchers develop more effective treatments. Moreover, the parallels between historical and modern malaria transmission underscore the importance of a global approach to disease control. In today's interconnected world, no region is isolated from the threat of malaria. International cooperation and coordinated efforts are essential to address the challenges posed by the disease. This includes strengthening surveillance systems, investing in research and development of new tools and treatments, and promoting public health education to raise awareness about malaria prevention. As we continue to battle malaria, the lessons learned from ancient DNA research can provide valuable guidance. By understanding how malaria spread and evolved in the past, we can better anticipate and respond to the challenges of the future. This research also highlights the importance of preserving and studying ancient human remains, as they hold the key to unlocking the mysteries of our past and improving our health in the present and future. Studying ancient diseases like malaria helps us understand these organisms better and develop more effective strategies against future outbreaks. Researchers can trace how diseases like malaria evolved and spread through history, providing valuable insights for current and future public health efforts. The integration of ancient DNA research with modern epidemiology is opening new frontiers in our understanding of infectious diseases. By examining the genetic traces left behind in ancient human remains, scientists can reconstruct the evolutionary history of pathogens and gain insights into the factors that influence their spread. This research is shedding light on the complex interplay between human societies, environmental changes, and disease dynamics. For example, ancient DNA studies have revealed how historical events, such as migrations, wars, and trade, played a crucial role in shaping the distribution of malaria and other diseases. These findings highlight the importance of considering social and cultural factors in disease ecology. By understanding the historical context of disease transmission, public health professionals can develop more targeted and effective interventions. The study of ancient diseases also provides a unique perspective on the long-term impacts of human activities on health and disease. The analysis of ancient malaria DNA from sites like Chocopani and Laguna de los Condores demonstrates how human mobility and environmental changes influence the spread of the disease over millennia. These insights can inform contemporary efforts to address the challenges posed by globalization, climate change, and urbanization. Furthermore, ancient DNA research is contributing to our understanding of the genetic diversity of pathogens. By analyzing the genomes of ancient malaria parasites, scientists can identify genetic variations that may have contributed to the emergence of drug resistance or changes in virulence. This knowledge can guide the development of new treatments and vaccines that are more effective against the diverse strains of malaria circulating today. The integration of ancient DNA research with modern molecular biology techniques is also advancing our understanding of host-pathogen interactions. By studying the genetic adaptations that allowed humans to survive in malaria-endemic regions, researchers can identify potential targets for new therapies. For instance, the genetic basis of resistance to malaria, as seen in sickle cell anemia and other blood disorders, provides valuable clues for developing interventions that can enhance immunity to the disease. In addition to its scientific contributions, ancient DNA research is fostering interdisciplinary collaborations and expanding the field of archaeogenetics. The study of ancient diseases requires expertise from multiple disciplines, including archaeology, anthropology, genetics, and bioinformatics. These collaborative efforts are driving innovations in research methods and technologies, leading to new discoveries and insights. The findings from ancient DNA research are also raising important ethical and cultural considerations. The study of human remains, particularly those of indigenous populations, requires careful consideration of ethical issues related to consent, repatriation, and the respectful treatment of ancestral remains. Engaging with descendant communities and respecting their perspectives and traditions is essential to conducting responsible and ethical research. As we continue to explore the mysteries of ancient diseases, it is crucial to recognize the broader implications of this research for public health and society. The insights gained from studying the past can inform our responses to current and future health challenges. By understanding the historical dynamics of disease transmission, we can develop more effective strategies to prevent and control infectious diseases, protect vulnerable populations, and improve global health. Thanks for watching. We hope you enjoyed this fascinating journey into the ancient origins of malaria. Don't forget to subscribe to our channel, like this video, and leave your comments below. See you in the next video.